Hi guys, sorry about that. I'm Daniel Rausch. This is Amanda Foster, Erica Shannon, Nicole Hurd, and Bryce Curtin. That's a great last name. We're the Missouri Miners. Our project is we're working on fuel cells, but another thing we really wanted to work on is we come from the middle of nowhere USA, from a conservative community, and we wanted to really work on community education and really getting out synthetic biology from where we're at. Amanda and Erica are going to talk about that for us. So here you go.
So like you said, we're the Missouri Miners. We're from Missouri University of Science and Technology, and our project is engineering an aerobic microbial fuel cell. Okay. So our team consists of about 20 students, all of which work on the team on a complete volunteer basis. We don't get any credit for working on the team or anything like that. We do all our work during the school year just because most of our students are not on campus during the summer. And we also recently became a student design team at our university. A little bit about Missouri S&T. It's located in Rolla, Missouri. You know, Missouri is in the middle of the U.S., in case you don't know. And Rolla is in the middle of Missouri, and it's a rural area. It's a very conservative community. We have about 7,000 students, most of which are engineering majors, and about 2% of our students are biology students, so it's a very small portion of campus. And another fun, interesting thing about our university is that we have a large St. Pat's tradition because St. Patrick is the patron saint of engineers. So as I said, we recently became the newest design team on campus at S&T, and that's a really big deal for us because student design teams are very well respected at our school. We have teams like solar car, solar house, concrete canoe, human-powered vehicle, and that's a really big thing on our campus, and we do very well on most of our competitions. So, and we also recently were recognized as an official student organization. Our team's only about 3 years old, so that was a good step. And we also have a lot of different majors on our team, which I think is really cool because it's not just bio people. We bring in chemical engineers and computer science and people from a lot of different disciplines, as you can see. As Daniel also mentioned, we're also trying to reach out to the community a little bit and spread awareness with synthetic biology. We also are involved in some community service projects, such as helping run the local science Olympiad competitions. And then Erica is going to tell you a little bit more about a survey we did related to public awareness.
Hi guys. Okay, so to determine the public opinion of synthetic biology on our campus community, we created a short survey of about 15 questions that measured response using the Likert scale. We distributed surveys in many different avenues and formats, and we had over 700 students, faculty, and staff respond. The data of our survey indicated that overall, individuals did not have a very deep understanding of the term synthetic biology. But once in the survey, when we provided them with a definition, we had over half the respondents indicate that they supported research in this area. We also wanted to compare the difference between the perceived connotations of the terms genetic engineering and synthetic biology. So our questions indicated that there is no significant difference between the two, or there's no perceived significant difference between the two on our campus, as you can see by the similarity of this data here. People did feel that there are risks associated with synthetic biology. However, over half the respondents did agree that it is beneficial to society. And then most individuals, we broke down synthetic biology into different components, environment, energy, and medicine. And as you can see by these numbers here, there's a lot of high support for synthetic biology, especially in engineering and medicine. However, the support dropped when we asked respondents about applications in food and agriculture. As you can see here, it's only about 50%. We can learn a lot from this survey. Our team learned that we can establish informationals to educate our campus community about synthetic biology. Next semester, we plan to hold open forums to discuss the implications and benefits of synthetic biology and the research. Now I would like to introduce you to Nicole, who will tell you guys about the heart and soul of our research.
Okay, so what we do, that's pretty big. That's a really bad angle. So we basically are making a microbial fuel cell, and some of you may know a little bit about that, I assume. Research has been conducted in this area before using naturally exoelectrogenic bacteria. These are bacteria that naturally produce electrons and shuttle them outside of their cell to reduce metals in the environment. And so when we're talking about these natural exoelectronic bacteria, we're talking about Geobacter, which is an anaerobic bacteria, and Shionella, which is a facultative anaerobe. So looking at this graph, you can see the top line there is Geobacter sulfurreduccens, and this is comparing voltage produced in similar microbial fuel cell environments. And so the top line is Geobacter sulfurreduccens. It has a fantastic electron transport system, and it creates a rather significant voltage there. And then Shionella is right underneath that. It has a significant voltage as well. And the benefit with Shionella is that you can use it in an aerobic environment, so you're not so worried about creating that tight security on your anoxic environment, which is somewhat difficult to maintain. And of course, our natural E. coli doesn't export electrons, so we have no voltage, basically. There's some, actually, but nothing significant. So our team thought, why not take this fantastic electron transport system out of Geobacter and put it into an aerobic bacteria, that we can have a nice electron transport system in an easy-to-use bacteria, more or less. So this is the electron transport mechanism for Geobacter. Basically, let's see, cytochromes inside the cell donate their electrons to these outer membrane cytochromes, B, E, S, and T, and then the electrons are shuttled off of them onto the pili, which then go to reduce the metals in the environment. Now, a lot of research has been done on this, and the pili are found to not be necessary for actually reducing the metals in the environment, so that's good, right? We can just take E. coli, which has these natural mechanisms already in its cell. Here are these inner membrane cytochromes and MAC-A and all this fancy stuff. And then we add in the outer membrane cytochromes, B, E, S, and T, which it doesn't have. And hey, boom, pow, we've got our fantastic electrogenic microbe. So that's what we did. We took those, we're taking those 4 outer membrane cytochrome genes from Geobacter and putting them into an E. coli plasmid and things like that. So our top plasmid there is from Geobacter. It contains all 4 of our cytochromes. And then we separated those into parts, individual plasmids. These are not actual BioBrick parts, but I do believe we submitted them to the registry last year, that they don't have the correct plasmids or anything. And then from there, we inserted them into the BioBrick part with the ribosome binding site. So each of these are now functional genes, theoretically. And then we have been cutting and ligating to produce this plasmid, which is the one we submitted this year, the BET together in one plasmid. Pretty soon we will be adding S in there too. We just haven't gotten that final step yet. And there were a lot of problems encountered
not necessarily problems, but interesting little tidbits here. As we grew our cultures, we realized that cultures that contained B and E together grew, it took 2 days for them to grow instead of just one. So we're thinking B and E put stress on the cells. And then when you add B, E, and T together, they grow like normal E. coli. And we've got, we digested the plasmids and ran them on a gel to determine that yes, we do have the gene there, but for some reason, that stress that's put on by B and E is reduced when you add T. And now I'm going to hand it over to Daniel, and he's going to actually have something else to say. So how do we know that these genes are actually producing the protein we want? We have to do cytochrome reduction assays. And so we did ferrous citrate reduction assays just last week, actually, and we haven't had time to do them again. But we tried it once because we know that it's important to verify our gene product, but unfortunately we weren't able to verify anything. It kind of turned up negative, but we only did it once, and so everybody knows in science you can fail once, but it could be really true. So I'm gonna turn it over to Daniel now for the real meat.
All right. Just to go back on those ferric citrate assays, when we were growing our bacteria, there's been some papers done that talk about the B gene, which is, I'm going to get back to this picture if I can get to it real quick. See how omcB is actually a transmembrane protein, and that's been found to be toxic to the cells occasionally. So when we were growing it with B, and when we had B and E together, our cells would grow really slow. So we were concerned about that. Maybe it was slowing down the growth and everything. But once we got B, E, and T together, they grew normal. So we were assuming that maybe they were getting together in a complex and getting throughout to the membrane and not harming the cells. When we were doing the reduction assays, we had a nice growth and everything. We put them in there, started going. Line wasn't going anywhere. So what we were thinking is maybe that the bacteria were starved, because occasionally, zillions of ideas. But we've got our reduction assays, and we're going to do some cytochrome staining just to make sure they're being produced. I'm concerned, and we've been concerned, that maybe they're not being produced. They might be selected out on these strains just because we were having low growth. So that's kind of that. I'm going to get on talking about the microbial fuel cells. So we have this neat exoelectrogenic bacteria, and we want to do something awesome with it. Our university has a lot of energy research being conducted. We have a nuclear reactor on campus. We have a coal fire plant, some hydrogen power things, and a wind power plant. So we wanted to take and integrate our biology department into these research projects. So along with our project, as we've been talking with Derek Lovely about microbial fuel cells. I stumbled upon a paper the other day about aerobic fuel cells research that was conducted at the Naval Research Labs. And we were kind of talking with Dr. Ringason about his concepts. So a microbial fuel cell is basically a battery that you continue to put fuel into, like glucose or sugar or something for the cells to eat. And then they export the electrons outside the cell. The electrons get captured by an anode material, which is usually a carbon felt, and it ends up running a circuit with a load. I have a beautiful picture right there. Good old animated GIFs. So anode chamber, we grow the bacteria. They attach to the carbon felt. They bring the electrons outside the cell. They run the load. You use titanium wire because it doesn't oxidize. And then you have a permeable membrane that you push hydrogen ions across. Usually it's a Nafion membrane or something of the sort. And then at the cathode chamber, it reduces oxygen like normal into water. We're just splitting the electron, stealing it for a little bit, and having some fun with it. Previous experiments, like I said, were conducted by Dr. Derek Lovely about Geobacter, and those are the genes we're using in our E. coli to give them a test. And the Naval Research Labs conducted some work on aerobic fuel cells to use as power sources in the water column, if they're using like stealth sensors, cool Navy things. So we talked to and contacted Dr. Ringason and asked them about their design, and they decided to shrink down the fuel cells. Traditional fuel cells are maybe the size of a laptop for testing, and they have a very small membrane chamber to push the hydrogen ions across. Well, what the Navy decided to do is they took that and they shrunk it down and used a special kind of carbon felt that has a huge surface area. This Mini-MFC design they use is actually the volume of 2 sugar cubes, and it's putting out 0.5 milliwatts. which doesn't seem like a lot, but once you start stacking up little sugar cubes together, it's not a lot of space and you're getting some power out of it. So we were using this, we're starting to test this idea, and one of the big things about our campus, because our biology department's so small, we have a lot of collaboration between other departments. The chemistry department in university, one of the professors, Dr. Nicholas Leventis, works in aerogels, and they've been doing some work on carbon aerogels as an anode material, and we're working with them and going to try that out. Mr. Bryce here is going to give you the rundown on that.
All right, so to further our progress in bacterial fuel cells, we plan to implement these carbon aerogels in both our anode and cathode. Aerogels are made by taking a chemical reaction that produces a gel and then replacing the solvent inside that gel with air by using supercritical CO2. So it maintains its porosity, and it has a high porosity, low density, and a high surface area. And with this high surface area, we hope to place more bacteria inside of it, which would then produce more electrons. And these carbon aerogels are also highly conductive, so they will be able to grab the electrons and shoot them out across the circuit. And here is an SEM image with a little bacteria to scale inside of it. So this is a 5-micron image of a carbon aerogel, and then little guy right there. So, and here to tell you how we keep track of it all is, again, Daniel.
So one of the other things about how our team is a volunteer organization, all our schedules are crazy. So some of us are working in other labs while we participate in iGEM, and some of us have crazy classes or applying for med school, and that takes up all the time. So what we decided to do, our university uses Apps for Education by Google, so we decided to upload our notebook online in a calendar format. It's on our wiki, you can go check it out. Some of our gel pictures are on there. The gel picture verifying our part got eaten by my dog, I'm sorry. So we've got this, it shows what we're doing on a day. You can click on the thing, you can see more details, and it'll show you everything we did that day. We can attach our gel pictures, show you what's going on. All it was just public knowledge so people could see what we were doing, other teams, and get experience and advice from us about how everything crazy was going. And on the scheduling part, we did the same thing and have some of our members here scheduled from a few weeks ago. Yeah, last week, or 2 weeks ago. So that's kind of how we did that. And talking about this, to us, iGEM, especially in our community, like we said, we're a small rural town, and we're conservative. So it's important for us to get that support from the community, especially with being a small team and collaborating with the entire university to try new projects. We've been working on the fuel cell thing and had to restart it a couple times because of lost information, lost genes in the freezer, that kind of thing. So part of it this year was to get everything organized, get ready to go. We've made a lot of progress with our project. Like we said, we're almost ready to go. We started the reduction assays. The concerns about having membrane proteins, I'm sorry. And we want to work on maybe in the future pushing this on, trying to get some more degradation things into the plasmid to use, I like to talk about Back to the Future where he takes banana peel and throws it in the reactor and the flux capacitor takes off. Maybe so we can use banana peels to power our little fuel cells, but that's Missouri Miners and our exoelectrogenic, what? Oh yeah, reference, I gotta say thank you or I'm gonna get killed. Sponsors, one of our previous members works at ExxonMobil now and they give us thing, and then Monsanto, they're from St. Louis. And we had a huge donation from Photo9, that's shameless plugs here. A lot of our support comes from our university with the energy research and the departments that support us. Dr. Leventis with the Aerogels, we're just starting that and feel like iGEM is a good springboard at our university to try other things. So we're gonna test out the new Aerogels, see how that turns out. References, Derek Lovely is awesome. And then questions this time.
I'm just wondering, do you foresee any problems of taking genes directly from Geobacter and put them, expressing them in E. coli?
Of course. I should probably steal a mic back here. One of the concerns is that since we're moving electrons right out the cell into an aerobic environment, that it would automatically just start reducing oxygen dissolved in the media, which has been shown, and in the paper I was referring to by the Navy, they did show it was a reduced electron potential. And I think Derek Lovely has a paper where he moved the E gene into E. coli, and it was showing some electric potential. So it seems like they've overcome, or it's not affected by the oxygen in the actual genes. But again, like I said, how some of the genes were actually toxic to the cells, that's been the big concern, is that maybe E. coli might not be the best model for this. Maybe trying something a little similar to Geobacter that can work in an aerobic environment. We also threw around putting them into Shmonella, which is facultative anaerobic, just to see if maybe the genes work a little better in Shmonella than E. coli.
So it's a great idea. I love the idea of making E. coli electric. There's potentially some good applications with that as well. But I just wanted to question you about, the sort of the pathway, like the choice of genes that you wanted to put together to make a complete electron transfer pathway. Could you go back to the diagram where you show the cross section of the periplasm? Yes, that's the one. So your constructs that you were building just have omcB, E, S, and T. But if I understand this diagram correctly, you want to have basically, a fire brigade, right, handing off electrons coming from the inner membrane, from the quinone pool, to this protein MAC-A, which then transfers electrons to a soluble paraplasmic protein, and then to this complex residing on the outer membrane. So what do you have in E. coli connecting your outer membrane complex with the inner membrane quinone pool?
That was part of the concern. I'm pretty sure B is what starts the siphoning from, since it's a transmembrane protein, but maybe in Geobacter it's not toxic and we're having problems in E. coli. So the MAC-A then kicks them out and it's in them, and we just pull them up through. S and T seem like they have a lot of analogous properties. And with the peelin, just to talk about that a little bit, it's been shown that it's not needed, but it does help with biofilm formation, and that is big on increasing electric potential. But it seems B is the key gene in getting them through the membrane to E.
Were you asking like how it got from the PPCA to omcB, that kind of thing?
How does it get from the inner membrane to the 4 proteins that you have residing on the outer membrane? The periplasm is about 150 angstroms, right? So you have to bridge that space somehow.
We kind of just assumed, perhaps not nicely, I suppose, we kind of assumed that it might just work. If it doesn't just work, we might have to consider that and figure out some sort of way. Do you know any way that?
You could add more proteins, but there may be E. coli proteins endogenously.
From the information, it seems like you just need those 4 cytochromes, and it has all the other things in between the membrane. That's just the differences, those outer membrane cytochromes. But, yeah, sorry.
It seems like you guys have a very unique opportunity, being from a rural area, to go out in that area and survey what people thought outside of an academic environment about synthetic biology. From your survey, was it all in the academic environment, or was any of it outside? Or
The survey was conducted on our campus, so the survey was an academic environment. However, we've had some exposure to outside our campus community. We were recently featured in our local newspaper, the Rolla Daily News, to try to get some exposure, to expose our research in synthetic biology to the community, but the survey results did not include responses from the community.
well, it seems like it sounds like something that'd be really interesting to learn how to communicate toward that population.
Yeah, we are in the future. We do plan to extend the survey to the community through our newspaper and other forums such as that. Do you have something?
I was just going to say, we're really interested in comparing also the results from just the people in our community versus our university's academic environment, and seeing if there's a large difference between the public opinion in those two areas. So that's definitely something we're going to look into.
Just to add a little bit to that, because it's a rural community and there's not a lot of telecommunication penetration, it's difficult to get a hold of the actual community. So we're working on part of that too, is just to getting it out there.
You guys have a wonderful idea. I really like it. Regarding the scale of growing this bacteria, you think if you grow the bacteria in large scale, like in a bioreactor or something, do you think there will be a potential of getting electricity of a generator of electricity, or you can't control the large scale growth of the electron or electricity producing bacteria?
We've scaled up the growth and it seems to be fine. A lot of it is growing it with the anode surface so they can attach in there. After we got all the parts together, it seems like they're starting to grow a little more, a little quicker. I want to do some more tests just to see if maybe that was a bad batch of cells we were using, competent cells, or what exactly was going on there. But in a bioreactor scale, it should be fine. That's what you're talking about, maybe putting them in and using like joint cultures, adding ours in with some other degrading things and see what happens.
Thank you.
I'll ask one too. So E. coli is not very good at making C-type cytochromes, especially under aerobic conditions. And many of these proteins are C-type cytochromes. And then the other issue is a lot of E. coli lab rat strains are not good at putting proteins on the outside of the cell, a process that typically uses type 2 secretion, although I'm not sure for these ones. So have you guys thought about using different E. coli hosts? And also, there are some tricks to get them to make more cytochromes.
I need to talk to you after this. Yeah, that was the thing is we were really constrained. We wanted to stick with the iGEM biobrick thing, using E. coli as a model organism. We took this project off for some seniors that graduated last year, and I've been punching holes through it and fixing things, trying to patch things up and where things went wrong. We're still finding problems, and I'm glad you brought that up because that was some of our concerns that have been here lately with the cells not growing and not actually getting any reduction in that assay. But we're still knocking our projects out here, so thank you.
Give all our teams another round of applause. Thank you.